Bismillah Rahman Rahim. Welcome back, friends. Welcome to another video lecture by Kami Biology. In this video lecture, we are going to talk about another organelles, and we call that Golgi body. So let me write the name. Golgi body. In this video lecture, we are actually going to talk about the introduction to Golgi body, structure of the Golgi body, and also function of the Golgi body as well. So these are this video lecture is actually obviously this video lecture are regarded with the cell. Now, Golgi body. First of all, let me tell you about the introduction of the Golgi body. Golgi body, we also call that Golgi complex, or we also call that Golgi apparatus as well. So first of all, the Golgi body, Golgi complex or Golgi apparatus was discovered by a scientist. We call that Camillo Golgi. So it was first of all discovered by Camillo Golgi in 1916. So first of all, it was discovered by Camillo Golgi in 1916. So that's why we call that Golgi bodies, Golgi complex or Golgi apparatus, because it was discovered by Camillo Golgi. So this Golgi body will present in all eukaryotic cell, but inside the fungi and ciliated protozoa, this Golgi body will be in a different shape, or we can say they have no regular shape. So this is a little bit introduction to Golgi bodies. So this is a little bit introduction to Golgi body. Now let's talk about the structure of the Golgi body. If we see the structure of the Golgi body, so Golgi body consists a plantated like structure, plantated sac-like structure. This plantated sac-like structure we call that cisternia, and each cisternia will be staked, means each cisternia will be present one above the other. So how actually these cisternia will look like? So let me draw the structure. So these are the cisternia, fine. And we also say that these cisternia are actually plantated, means that one thing more, it is plantated, and one thing more, it is staked, means it is present one above to each other, one above to each other. So this is a little bit about the cisternia. What we call that? We call that cisternia. Yeah. Now, the thickness of each cisternia, or first of all, inside the cell, or we can say inside the Golgi body of the eukaryotic cell, the cisternia amount will be almost the cisternia amount will be 4 to 8. So each Golgi body contain 4 to 8 cisternia, or also they contain more than 4 to 8 cisternia as well. And so it is depend upon the size of the cell. Or now if we see the thickness of this cisternia, so the thickness of the cisternia is almost 15 to 20 nanometer. So the thickness of this cisternia will be 15 to 20 nanometer, and each cisternia will be separated from each other, and the separation or the space between each cisternia will be 20 to 30 nanometer. 20 to 30 nanometer there will be a space, or we can say they will be separated from each other. And now these group, if we see this is the cisternia, these are also the cisternia. So we can say this group of cisternia are called what? These group of cisternia are called dectisomes. Dectisome. So this is a little bit about the cisternia and dectisomes. Now at the age of cisternia, if we see at the ages of cisternia, it contain a tubules-like structure, means this tubules-like structures. So it contain a tubules-like structures. Fine. So these tubules-like structure we called it contained a tubes-like structure, we called it tubes. or we also call it tubules. So these are what? These are the tubules. Moreover, at the edge of this cisterna, it also contain what? It also contain the secretory vesicles, as I draw with the green marker. So it also contain the secretory vesicles. These are what? These are the secretory vesicles. So this is about the secretory vesicles. Now the diameter of each secretory vesicle will be 2200 nanometer. So their diameter will be 2200 nanometer. Now moreover, this cisterna will also contain two faces. How many faces? They contain two faces. This upper face we call that what? This upper face we call that trans phase. Trans, or we also call it maturing phase. Maturing phase. Now, what are the main function of this trans or maturing phase? The main function of this trans or maturing phase, they are responsible for the formation of this secretory vesicle, as I draw with the green marker. So actually they are responsible for the formation of these secretory vesicles, and this means this trans or maturing phase will also contain enzyme as well. They also contain another phase which are present at the lower end or at the bottom. So let me draw with the black marker. So these are another phase. We call that cis or forming phase. We call it cis or forming phase. Now this cis or forming phase is actually associated with the endoplasmic reticulum, if you remember endoplasmic reticulum. So that's all about the Golgi bodies, the structure of the Golgi bodies. Now let's talk about the function of the Golgi body, means the vesicles which are synthesized inside the endoplasmic reticulum. These vesicles are actually contain the proteins, and once they receive the vesicle, then inside the Golgi bodies, then what actually happen inside the Golgi body, they can convert the protein into a useful shape. For example, this is a vesicles, let's suppose, and they contain what? They contain the proteins molecules. Now these are the proteins. Now these vesicles are received by this Golgi body. What actually Golgi body do? They will customize this protein into a different shape, or they will customize the protein into a useful shape. For example, they will customize the protein just like in this shape. Once they customize the protein in this shape, then the cell will use this protein for the energy. Moreover, inside the Golgi body, what actually happens? They also add another molecules, we call that lipids, or we can say another molecule, we call that these one. Let's suppose this red one represents carbohydrates, which the cell needed. So they can sometimes they attach the lipid, or sometimes they attach the carbohydrates which the cell needed. Fine. So this is about the function. Now the next function of this Golgi bodies, they can collect the waste material, and once they collect the waste material, then they expel out these waste material in the form of secretory vesicles. Means this secretory vesicle are actually contain what? Waste material, and these waste material are actually expelled out from this Golgi bodies, and then these waste material will be shift to the cell membrane. From the cell membrane, then these secretory vesicles will be these secretory vesicles will be burst. So from the cell membrane, these secretory vesicles will be burst. We can also say that these Golgi bodies are also involved for the development of the cell membrane as well. So inside the fungi, if we see the fungi cell wall or growth, the fungi cell wall or growth is possible by this Golgi body. If the Golgi body function properly, so the fungi cell wall also develop. So we can say that this Golgi body is actually also responsible for the development of the cell membrane or the cell wall. Fine. So that's all about the Golgi bodies. I hope this video is helpful. If you like this video, please hit the like button, share this video. And if you have questions related with this topic, then write it into a comment. Inshallah, I will give you answer as soon as possible. So please like the video, share the video, and subscribe the channel to get more updates of the. So please like the video, share the video, and also subscribe the channel to get more updates of the cell. Thank you so much.